Welcome to Lecture 21, where we will go over neurons and their role in the nervous system. Once the action potential passes down the axon, it will reach the synapse. There are two main types of synapses that can be found, electrical and chemical. At an electrical synapse, gap junctions will be present to allow for the electrical current to flow. This type of synapse is typical between neurons. These synapses are very important in synchronizing neuronal activity to allow for rapid responses. These synapses are present in giant axons, as well as in vertebrate hearts and brains. Chemical synapses, on the other hand, involve the release of neurotransmitters at the synapse to cause a response in the target cell. In these types of synapses, the action potential will cause a depolarization of the neuron plasma membrane. This will cause voltage-gated calcium channels to open, which will lead to an influx of calcium into the neuron. This will cause the synaptic vesicles that contain the neurotransmitter to fuse to the membrane. The neurotransmitter will then be released into the synapse, where it can then bind to receptors on the target cell membrane. This diagram depicts the pathway in a chemical synapse. As the action potential travels down the axon, note that we have synaptic vesicles that are present within the neuron that contain the neurotransmitter. Once the action potential reaches the synaptic cleft, this will then open the voltage-gated calcium channels. As calcium rushes into the neuron, it will cause the vesicles to fuse to the membrane at the synaptic cleft. This will release the neurotransmitter into the synaptic cleft, where it can then reach the receptors on the target cell membrane. At most chemical synapses, the binding of the neurotransmitter to the target cell membrane causes ligand-gated ion channels to open or close. This results in a postsynaptic potential that can either be excitatory or inhibitory. If the potential is excitatory, this is caused as sodium and potassium channels open. This will cause a depolarization event that moves the membrane potential towards the threshold, thus it is getting less negative. In an inhibitory potential, potassium and chloride channels open. This will cause a hyperpolarization event that moves the membrane potential away from the threshold, thus makes it more negative. In some circumstances, these potentials can act in a summative manner. This means that they have an additive effect that can produce larger postsynaptic potentials. Overall, it is the balance between these excitatory and inhibitory postsynaptic potentials that helps to integrate information within the nervous system. We're now going to take a look at another event at the synapse, the termination or modulation of a signal. Once a response is triggered, the synapse must return to its initial state. This is called termination. In a termination response, the neurotransmitters will be removed from the synapse. This can be done through the enzymatic degradation of the neurotransmitter or by reuptaking the neurotransmitter and repackaging it within the neuron. In a modulatory response, this may involve the neurotransmitter binding to a G-protein coupled receptor. This causes the activation of signal transduction pathways within the target cell. Many times, the modulation response occurs along with the typical response, involving ligand-gated ion channels. There are many different types of neurotransmitters that can be present within neurons. However, it's the action of the neurotransmitter binding to its receptor that will determine the ultimate response in a target cell. Take a look at the diagram to the left to understand the role of these various neurotransmitters. Common neurotransmitters involve acetylcholine. Acetylcholine is the basic neurotransmitter that is responsible for stimulating muscles, memory formation, and learning, which is very important and becomes pronounced in vertebrates. Amino acids can also work as neurotransmitters. These include glutamate, glycine, gamma-aminobutyric acid, or GABA. Biogenic amines are synthesized from their base amino acids. These neurotransmitters include dopamine, serotonin, as well as norepinephrine. Neuropeptides consist of short chains of amino acids that act as neurotransmitters. These include, for instance, P as well as endorphins. And finally, gases can act as neurotransmitters at the synapse. This includes nitrous oxide and carbon monoxide. We're now going to take a look at how neurons are organized within body systems. Complex activities such as speech, cognition, and memory formation are carried out in a highly coordinated information processing pathway in the body. In general, a nervous system consists of neuronal circuits and their supporting cells, and these systems can vary in complexity. Simple organisms like Hydra and cnidarians have a nerve net of interconnected neurons that helps to spread information throughout the body. More complex organisms like the starfish may have their axons bundled into nerves that extend into various body regions. This can be seen in the presence of a nerve ring as well as the radial nerves that extend into the arms of the starfish. Many bilateral animals show what is termed cephalization. This occurs when there is an aggregation of sensory neurons and interneurons at the anterior end of the animal. In addition, nerves will extend posteriorly to the rest of the body. And finally, animals that need to integrate information may have specialized central and peripheral nervous systems, or they may have ganglia present throughout the body to help transmit information. In general, the vertebrate nervous system is organized into two main divisions, the CNS and PNS. The CNS, or central nervous system, develops from the dorsal hollow nerve cord. This will give rise to the brain and the spinal cord. The peripheral nervous system includes cranial and spinal nerves, as well as ganglia, that help to receive and relay information to and from the central nervous system. The central nervous system includes the brain and the spinal cord. These tissues are nourished by the cerebrospinal fluid, or CSF. The CSF is produced by the choroid plexus that lays within the ventricles of the brain. This will bring nutrients and hormones to the brain and also help eliminate waste as it drains into the main veins of the head. Gray matter consists of cell bodies of neurons, while the white matter consists of the axons. In the brain, gray matter forms the outer layer, while the white matter forms the inner layer. However, this is reversed in the spinal cord. Note that in the spinal cord, the gray matter is considered to be internal, while the white matter is considered to be external. This means that the cell bodies in the spinal cord are interior, while the axons are exterior. Reflexes involve autonomic responses for certain stimuli. Reflexes typically involve quick responses that help to protect the body. Reflexes are controlled by single nerve circuits that involve a sensory neuron, interneuron, and motor neuron. This information is integrated and processed within the spinal cord, which allows for a rapid response without having to process the information within the brain. Note the presence of a typical reflex response here. Sensory neurons will be present in order to detect the stimulus. This will convey that information through action potentials into the spinal cord. Interneurons may cause the inhibition or activation of certain muscles. Motor neurons will then convey that signal to the target tissue to cause contractions. The peripheral nervous system is responsible for carrying information to and from the central nervous system. This information is carried through afferent and efferent neurons. Afferent neurons will bring information to the CNS, and efferent neurons will bring the information from the CNS. The peripheral nervous system is divided up into two systems: the motor system that controls skeletal muscle, and the autonomic nervous system. The autonomic nervous system is broken down into the sympathetic and parasympathetic systems. These typically counteract each other to help control smooth muscle, cardiac muscle, as well as the action of glands. The enteric nervous system is responsible for controlling the digestive tract, pancreas, as well as gallbladder. This diagram represents the organization and integration of information between the CNS, central nervous system, and peripheral nervous system. Note that internal and external stimuli will be detected by sensory receptors. Afferent neurons will then bring this information to the central nervous system for processing. Once the information is processed, efferent neurons will carry this information to either the motor system or the autonomic nervous system. The parasympathetic and sympathetic divisions control different actions within the body. The parasympathetic division controls the rest and digest response, whereas the sympathetic division controls the fight or flight response. In the parasympathetic nervous system, neurons will exit the CNS at the base of the brain and spinal cord. These neurons will then form synapses with ganglia near the target organ. Preganglionic neurons have cell bodies that are present within the CNS, and they typically involve the release of acetylcholine. Postganglionic neurons will release acetylcholine as well. In the sympathetic division, there are major fundamental differences. These involve the exit of the neurons from the CNS midway along the spinal cord, rather than at the base of the spinal cord that we see in the parasympathetic division. When the neurons from the sympathetic division exit the CNS, they will form synapses with ganglia near the spinal cord, as opposed to near the target organ in parasympathetic nervous system. Preganglionic neuron in the sympathetic division have cell bodies that are located in the CNS, and they typically release acetylcholine, which is similar to what we see in the parasympathetic division. However, in the sympathetic nervous system, postganglionic neurons will release norepinephrine. Take a look at the diagram to the right to understand the differences between the parasympathetic and sympathetic division. Most of these pathways involve two neurons that connect the CNS or spinal cord to the target organ. However, note that the synapses are present near the target tissue in the parasympathetic division, whereas they are closer to the spinal cord in the sympathetic division. Also note the difference in the neurons leaving the parasympathetic versus sympathetic division. The parasympathetic neurons leave the base of the skull, whereas the sympathetic neurons leave throughout the spinal cord. In general, the parasympathetic and sympathetic divisions will counteract each other on target organs. For instance, the parasympathetic division will result in the slowing of the heart contractions, whereas the sympathetic division will increase the heart rate. There are also differences in the neurotransmitters that are present within the peripheral nervous system. These will allow for varied effects on target organs. The motor system neuron, as depicted in the diagram to the left, consists of single neuron circuits that allow for rapid responses. The autonomic nervous system below has both pre- and post-ganglionic neurons, as well as different ganglia found throughout the body to help coordinate responses. These are very important in maintaining homeostatic control. Acetylcholine and norepinephrine are both important neurotransmitters that act within the peripheral nervous system. The balance between acetylcholine and norepinephrine can cause varied responses within their target tissues. Glial cells are also important supportive cells that occur within vertebrate nervous systems. These glial cells can be present throughout the nervous systems, CNS as well as PNS. These glial cells include ependymal cells that line the ventricles of the brains and help circulate CSF, astrocytes that help to form the blood-brain barrier, oligodendrocytes that myelinate the axons in the CNS, Schwann cells that myelinate the axons in the PNS, and microglia that act as immune cells within the peripheral nervous system. Glial cells are also present during embryonic development. Radial glia during development help to form tracks for newly formed neurons as they migrate away from the neural tube. These will help forge the path for neurons to extend, which will eventually lay the foundation for the formation of the peripheral nervous system.